The National Institute of Neurological Disorders and Stroke is making targeted investments in epilepsy research to address some of the most difficult challenges in the field. These investments have been formalized in a program called the Epilepsy Centers Without Walls, and the topics of the Centers Without Walls have really emerged from conversations with everyone in the epilepsy research community. Scientists, physicians, other healthcare providers, patient advocates, and funders of research. And it's clear from those conversations that some of the most pressing and difficult questions are, what are the causes of epilepsy? What are the reasons why the rates of sudden unexpected death are so high in patients with epilepsy? The first center was awarded to the Epi4K Consortium with the goal of better understanding the genetic contributions to human epilepsy. And their early results have identified new genes that contribute to some of the rare and catastrophic epilepsies in children. The second Center Without Walls will focus on understanding mechanisms and potential prevention strategies for sudden unexpected death in epilepsy.
We don't know what causes SUDEP, although there are now some theories that will be explored in a multidisciplinary Center Without Walls called the Center for SUDEP Research. This center brings together 12 investigators from 11 institutions around the country, around the United States and the UK, to examine the brainstem areas and other brain areas that might be involved in controlling respiration and heart rate, that when there's seizures and dysfunction in these areas, ultimately lead to sudden death.
In just the last decade, clinical research and peer-reviewed literature on SUDEP has seen a welcome and exponential increase. This marks the efforts of a diverse group of people, tireless campaigners from patient advocacy foundations, concerned parents of patients, clinical care professionals, researchers, and funding bodies. With the investment now being made by the NIH into SUDEP research, we have a real chance at defining individual risk, pinpointing those at a special risk, developing preventive strategies, and reducing overall epilepsy mortality. From recent research, we know that some patients with intractable epilepsy have characteristic EEG or brainwave signatures that mark high risk for SUDEP. We call this prolonged post-ictal EEG suppression. Recent research has also revealed that patients who succumb to SUDEP, in addition to these EEG signatures, also have strange breathing and heart rate patterns that lead to the terminal event. Further, failure of blood pressure homeostasis has been shown to occur in some patients. However, we don't know how these brainwave signatures correlate with critical malfunction of breathing, blood pressure, and cardiac function. The translational and reverse translational underpinnings to these are going to be genetic influences, brain neurotransmitters, and ion channels, areas in which promising progress has also been made in SUDEP research. Already, we've begun to make inroads into identifying markers of high risk using brain imaging techniques. Certain parts of the brain in long-standing seizure patients, as a result of accrued seizure-induced damage, may show structural and/or functional changes that modify cardiac and breathing responses to seizures. We now have an opportunity to bring all this apparently disparate research from the best minds in the field, from both clinical and basic science arenas, to cross-fertilize each other's domains and to bring to fruition complete knowledge of the agonal processes that drive SUDEP. In so doing, we hope, as all of us in the epilepsy field do, to bring hope, comfort, and confidence to our patients and their loved ones who don't deserve the fear, the uncertainty, and the catastrophe that the phenomenon of SUDEP brings.
The NIH has funded an extraordinary project now to try to help us uncover some of the mechanisms and perhaps risk predictors. So the plan is to harness a clinical network of 10 epilepsy monitoring units to identify and study individuals with seizures complicated by very abnormal breathing and heart rate problems. Then we'll collect samples of DNA and skin fibroblasts. From the DNA, we can detect genetic mutations, and from the stem cells in the skin, incredibly, we can transform them into several types of brain cells, both excitatory and inhibitory, and heart cells, and study their properties for defects. While this clinical detective work is going on, we have a very focused program in basic science laboratories to build and study genetically engineered mouse models of these human defects to examine the underlying physiology, and to learn how single gene defects can produce the severe form of epilepsy complicated by cardiorespiratory instability. So we're just hoping that this unique team of expert investigators, both clinical and basic, can come together over the next few years in this program and put together a risk calculator that we can offer to clinicians.